gonna study. Hopefully I'll get all this studying over with and get caught up. Okay. Starting off with physiology and histology. Got a quiz tomorrow. And then I have an immunology exam tomorrow. So. Okay, back to. We have osmolality, osmolarity. Again, they're used somewhat synonymously. But then there's osmotic drivers. In the extracellular fluid, sodium and chloride make up about 80% of total osmolarity. Sodium and chloride in the ECF. 80%. In the ICF, potassium makes up about 50% of total osmolarity. Osmosis is the net movement of water across a membrane due to a concentration difference for water. Simply put, the water is following particles. Osmotic pressure is exerted by particles in a solution, and it is determined by the number of particles per unit of fluid, not the mass of the particles. It's determined by the number of particles, not the mass. Hydrostatic pressure. This is pressure differences. For example, capillary pressure is 20 millimeters of mercury higher inside than outside the capillaries. Hydrostatic pressure drives blood out of the capillaries. Okay, moving on to solute permeability and tonicity. There's something called a reflection coefficient, which is a number between 0 and 1, describing the ease with which a solute permeates a membrane. If the number is 1, then solute is impermeable. This is something like serum proteins. And it creates osmotic pressure. Now, if the value is closer to 0, or if it is 0, hey guys, what's up? Then the solute is completely permeable. For example, urea. If you've got urea, it has no osmotic effect. It just goes in and out of the cell, no problem. Okay. Then we have something called an effective osmole. An effective osmole is a solute that does not cross a membrane easily and thus creates an osmotic force for water. Plasma proteins are effective osmoles for the vascular compartment. That's an example again. What does that mean? So you've got particles over here. An effective osmole is something that stays over here, and yet water can move back and forth. If you've got a selectively permeable membrane here, the effective osmole cannot cross, but water can. Water can cross easily. Okay. And what that does is the water goes to the particles and allows osmosis to occur. Tonicity is the effect of the solution on cell volume. So you've got isotonic solutions. These cause no change in cell volume. If an animal were dehydrated and I needed to give it fluid very quickly, you could administer saline intravenously or subcutaneously. That is an example of an isotonic solution. A solution that is adapted to the body's tonicity. aka the body's solute concentration. Then there's hypotonic, which causes water to enter the cell and it causes cell swelling. Then there's hypertonic, which causes water to leave the cell and it causes cell shrinkage. Like, for example, a crenated, not cremated, crenated, or I don't know how you say that, crenated red blood cell. I don't know what crenation means. Let me look it up. Probably shrunken. It says crenation in botany and zoology describes an object's shape, especially a leaf or shell, as being round-toothed or having a scalloped edge. The descriptor can apply to objects of different types. Okay. So yeah, it just gets shrunken, but maybe it starts to look a little spiky. Something like that, I guess. Okay, moving on. Fluid interfaces and regulation. So you've got fluid barriers. One of the most common fluid barriers is the cell membrane. This is the interface between intracellular fluid and extracellular fluid, ICF and ECF. Water moves across and it's driven only by osmotic force during that process. Next, we've got a capillary wall. That's another example of a fluid barrier. This is an interface between interstitial fluid and plasma. Water moves across and it's driven by both hydrostatic and osmotic forces. Again, the example you could use here is plasma proteins. Proteins in the plasma. Next, we have ECF regulation, another example of fluid interfaces and regulation, which is extracellular fluid regulation. The priority of this is that cell function is going to be more immediately affected by changes in ECF osmolality than ECF volume. All of these changes to fluid balance begin in the ECF, which then impacts the ICF. And then there's something called the Darrow-Yannett diagram rules. Basically, ICF volume is determined by ECF osmolarity. ECF volume is determined by whole body sodium chloride content. I'd show you a picture, but I can't because I don't own any of the sources. I don't own any of the materials. Next, and finally for ECF regulation, we have regulatory systems. Now there's two ways or two systems outlined here. There's renin angiotensin aldosterone system, RAAS, aldosterone. Well, RAAS, also just called aldosterone, primarily regulates ECF volume, which is ECFV, ECF volume. And aldosterone increases sodium and subsequent water reabsorption in renal principal cells, kidney principal cells, but does not change extracellular sodium concentration. Okay, and then we have the second one, which is antidiuretic hormone, ADH, or there's also arginine vasopressin. Diuretics are something that make you pee, and so antidiuretic makes you not pee. Why? Because it's taking the water out of your cells and sending them to the kidneys. Let me look that up, make sure I'm not getting that wrong. Diuretics, commonly known as water pills, are medications that help the body eliminate excess fluid and salt by increasing urine production. Yeah, so they cause your body to excrete more water via the urine, not sweat or anything like that. And anyway, this ADH, it primarily regulates extracellular osmolality. It's released when hypothalamic osmoreceptors detect an increase in plasma sodium, which is ECF osmolarity. It increases water permeability and reabsorption, which leads to small amounts of concentrated urine. Okay. Next, moving on to clinical fluid abnormalities and cell volume regulation. First, we have hyponatremia. So, hypo meaning not enough, natreme. If you start off with the beginning of that, Na, well, there's sodium. Na, it's the elemental symbol for sodium. Hyponatremia is a lack of salt, pretty much. And so in this case, it's cell swelling because there's not enough salt inside the cell and there's more outside the cell. So water, well, hold on. This is different from hypotonicity. Hyponatremia is going to, it causes cell swelling because it's also, I don't know, that's weird. Hyponatremia must also be hypotonicity. Okay, but it's defined as a plasma sodium of less than 142 MEq per liter. Not sure what the MEq means yet. Okay. Some causes of hyponatremia are loss of salt or excess water. These can be due to diarrhea, vomiting, overuse of diuretics, Addison's disease, decreased aldosterone, and SIADH, which leads to excess water retention. Some consequences of hyponatremia are cell swelling, especially profound effects on the brain, which can be edema, headaches, seizures, coma, or death if the plasma sodium is less than 120 millimoles per liter. There are ways to compensate this, though, like regulatory volume decrease, or RVD. Basically, if hyponatremia is not sudden, cells can adapt. It takes about 1 to 3 hours, and they do this by deliberately dumping osmolytes like sodium chloride, potassium, organic solutes like glutamate to lose water and reduce swelling. And there's ways we can treat it as well. Hypoosmotic sodium chloride or dextrose is one way. You administer it slowly, because if you administer something like that too quick, you can cause osmotic injury to your neurons. So that's not a good thing. Next, we have hypernatremia, cell shrinking. So this is defined as a plasma sodium of greater than 160 MEq per liter, caused by a loss of water from the ECF or excess sodium, which can be due to prolonged heavy exercise, inability to secrete ADH, large amounts of dilute urine in diabetes insipidus, hypothalamic lesions, paintballs, don't eat paintballs, and homemade Play-Doh. Don't eat Play-Doh. Can lead to cell shrinking, intense thirst, stimulation of ADH, confusion, muscle twitching, spasms, and intracranial, not intracranial hemorrhage, bleeding inside the brain cells, or at least inside the cranium. The body compensates this by a regulatory volume increase, which is a short-term thing. Cells can gain inorganic osmolytes as a result, which are things like sodium, chloride, potassium. And in the long term, they can gain organic solutes like sugar alcohols, amino acids, and methylamines. And it's found that hypernatremia actually is associated with a higher case fatality rate than hyponatremia in dogs and cats. Hypernatremia, however, is less common because it causes intense thirst and increased ADH secretion. You know, you're just going to drink water if you feel thirsty. Again, there's always a way to treat these things. However, you can just give hypoosmotic saline, which is going to be administered slowly due to cell adaptation. But yeah, it's treatable. All of these are treatable. Then there's something called edema, which is excess fluid in tissues. There's two types: intracellular or extracellular edema, in the cell or outside of the cell. Intracellular edema is caused by hyponatremia or depression of cell metabolism, that's when ischemia causes ionic pumps to fail. Then it can also be caused by lack of nutrition or protein losing enteropathy, which is also called hypoalbuminemia. And that's intracellular edema. An example of this is brain edema. Cytotoxic edema is a type of brain edema. There's all sorts of different types, which can be caused by loss of oxygen or sodium accumulation. Then there's vasogenic edema, which can be caused by the loss of the blood-brain barrier. Very important physiological thing. And it can also be caused by protein extravasation. Next, the causes of extracellular edema are abnormal leakage from plasma to interstitial space, which is the most common form of extracellular edema, or failure of lymphatics. The lymphatic system fails. And that's called lymphedema, right? Probably heard of that one before. There are some leakage factors in extracellular edema, like increases to capillary permeability, increases to hydrostatic pressure, a decreased plasma osmotic pressure, and by the way, that decreased plasma osmotic pressure is a result of decreased plasma proteins. And then there are some safety factors, ways to prevent extracellular edema, like low compliance in the interstitium. What is compliance? Well, a high compliance means the tissue moves more freely. Your lungs are very compliant. Your skin, not so much, but still somewhat. Your bones, almost not at all. That's what compliance is. So, Again, these safety factors, one of those is, again, is low compliance. Talked about that. And that happens because your interstitial fluid pressure is in a negative range, and you're going to have fluid trapped in the proteoglycan gel. Which is part of that. And then we have another way to prevent it is a wide range of lymph flow. Your lymph is constantly flowing, and it can increase 10 to 50 times. And then finally, a wash down of interstitial fluid protein concentration. What this is it's the lymph removing filtered protein and reducing interstitial colloid osmotic pressure. Okay, moving on to blood components. Plasma versus serum. What's the difference? Well, plasma is the fluid component with an anticoagulant, including clotting factors. Serum is the fluid without clotting factors. So plasma clots, serum does not. Serum has no anticoagulant needed or used because it doesn't have clotting factors. Plasma clots, serum does not, unless you use a coagulant. Plasma is where you would use anticoagulant. Plasma clots, that's why you use anticoagulant. Serum does not clot, so you don't need to use anticoagulant. Okay. Then there's erythrocytes, which are another fancy word for red blood cells, RBCs. These are biconcave disc-shaped cells, and they're shaped like that because they provide a large surface area for oxygen diffusion while still being able to fit through capillaries and stuff like that. They contain hemoglobin and carbonic anhydrase, which is an acid-base buffer. And they are anucleate. They don't have a nucleus when they're mature in dogs, cats, and other mammals. The lifespan, however, of these cells varies by the species. For example, dogs' RBCs last 120 days, and cat RBCs last 70 days. Okay, there's something called erythropoiesis. This is a fancy term for RBC generation in the bone marrow. It is stimulated by EPO from the kidneys when oxygenation decreases. For example, your oxygenation can decrease whenever you've got a low blood volume, whenever you've got anemia, or when you've got low hemoglobin. There are some ways to diagnose these things, diagnostics you can run, things you can notice, like jaundice, called sometimes icterus. It results from abnormal hemolysis, which produces bilirubin, and then the bilirubin stays in your body. Macrocytic anemia, which is immature red blood cells, and circulating reticulocytes or normoblasts indicate hemorrhage. Those are just a couple things you can notice that would, those are problems with red blood cells. Okay, then we have leukocytes, another word for white blood cells. These are things like neutrophils, eosinophils, basophils, lymphocytes, and monocytes. Leukocytes are formed in the bone marrow, and they don't last very long. They only live about 4 to 8 hours in the blood, but 4 to 5 days in tissues. Then we have platelets, which are also called thrombocytes. These are cell fragments of megakaryocytes, and they're important in hemostasis. These are also anucleate. They don't have nuclei. Their production is controlled by thrombopoietin from the liver and the kidneys. Okay, moving on to vasculature and smooth muscle. It's good to know the vascular structure and pressure. There are layers to blood vessels. From outside to the inside, we have the tunica externa, also called the tunica adventitia. It is made of collagen and it is for support or anchoring. Then we have the tunica media. Think media. What is media? Well, it kind of sounds like middle, doesn't it? That's exactly what it means in Latin. It's the middle tunic, right? The middle covering. And this is actually smooth muscle and elastic fibers. This controls the size of the lumen, and it is the thickest in arteries. Tunica media is going to be thicker in arteries. Finally, we have tunica intima. Think intima, kind of sounds like intimate, close, right? The closest to the center layer. Otherwise, you might call it an endothelium. It's the inner layer. Okay, then moving a little bit more specific, we've got arteries, which are high pressure. These are thicker, more muscular walls because they've got to handle that pressure. They've also got a thinner lumen, though. So the wall, the wall might be this thick, but the lumen is only like this thick. Whereas in a vein, maybe the wall's like this thick, but it's a little bit bigger, less pressure. And arteries are less compliant as well, meaning they don't move as easily as veins do. And they hold their shape better. Large arteries, like the aorta, are elastic reservoirs due to the Windkessel effect. Veins are low pressure. Thinner, they've got thinner walls as a result, because they don't need all the strength to hold the high pressure. They've got a larger lumen to move more blood. They are more compliant. They are a blood reservoir, and they contain valves for one-way flow. Those valves are there to prevent backflow. Next, we have arterioles, which are also called resistance vessels. This is where the greatest pressure drop occurs. They change resistance primarily by changing their radius. Resistance is inversely proportional to the radius to the fourth power. And this is Passai's equation that determines that. Think about it. If you have a water hose, you're watering the flowers, and you put your thumb over the top, what happens to the water? It shoots with more force, right? That's because the resistance increased greatly due to a decrease in radius from my thumb being held over the water exit. It's the same in your arteries. If you get clogged arteries, your blood pressure goes up. because your arteries are clogged up. You got a smaller diameter, radius, and so a greater resistance and a greater pressure. Sorry, my nose is stuffed today. Okay. Next, talking about capillary structure and exchange. So the capillary wall is a unicellular layer of endothelial cells. Exchange occurs primarily via diffusion through intercellular clefts. These are slit pores about 6 to 7 angstroms wide, or it can happen through plasmalymal vesicles, also called caveoli. Some permeability factors of capillaries are the size. If it's a smaller capillary, it's got faster diffusion. And solubility in water. Uncharged or lipid-soluble things diffuse easily, while charged or polar molecules are slowed or prevented. Proteins barely diffuse. There's different types of capillaries, though. There are continuous capillaries. These are the most common type. And they have junctional complexes. Then there's fenestrated capillaries, which allow small molecules and proteins through. And then there's discontinuous capillaries, which have large gaps, period. And free transfer of proteins and RBCs, and these are found in the liver. Finally, the interstitium of a capillary contains interstitial fluid, collagen fiber bundles, and proteoglycan filaments. And this gives it a gel-like consistency. I'm going to take a break, and I'll be back in like 5 minutes. Thank you for watching. See you in the next one. Appreciate y'all for watching.